Hello all. I'm Mohit Kumar, working as an assistant professor in IIMT College of Pharmacy, Greater Noida. Today I'm going to present my lecture on topic alkaloids. As we studied in pharmacognosy, like the drugs we like the fruit drugs and we phytochemicals we obtain from the drugs. We alkaloids is one of the phytochemicals we obtain from drugs. We basically study in the lecture about alkaloids, its physical and chemical properties, its classification, uses, and chemical tests like identification and confirmatory tests. So starting from the introduction part. So first, the definition. So basically, the term alkaloid was first introduced by the German scientist Carl F.W. Missner in 1819. Alkaloid are alkali-like. He told us like alkaloid are alkali-like. So basically, he told that alkaloid are basic in nature and contain a one nitrogen group. These are the basic definition of alkaloid. So Leidenberg defined alkaloids. He gave the proper definition of alkaloids. He told that group of naturally occurring organic compounds which are basic in nature contain one or more nitrogen atom, normally of heterocyclic nature, and possess specific psychological action on human or animal. So from this definition, we can conclude that like alkaloids are organic compounds in which contain one or more nitrogen atom in a heterocyclic ring or nature, and they have physiological effect on human or animals. So now coming to the distribution, like where we found these alkaloids. So basically, these are found in higher plants, like in angiosperms, and in the plants which belong to the families like Apocynaceae, Rubiaceae, Solanaceae, Pepperovaceae, etc. So the plants which come in these families also contain alkaloids. So there are in some plants there are specific parts from where alkaloids are derived, and in some the whole plants contain alkaloids. So we can now we will study the examples. So the alkaloids are procured from various parts of the plant. Like in Datura plant, all parts contain alkaloids. In aerial part of ephedra, alkaloid is obtained, and the phytochemical which we obtain is ephedrine. Vinca is the most important drug, like it is an anti-cancer drug, and basically two phytochemical, or we could say active constituents are obtained, that is vincristine and vinblastine. And in Vinca, entire plant alkaloids are found. In the leaves of tea, caffeine, and in roots of Rauwolfia, reserpine is the active constituent, and these are found in the roots of Rauwolfia. And bark of Shinkona. This was also one of the most important drug, like Shinkona quinine is used in the treatment of malaria. And in seeds of Nux vomica, in fruit of black pepper, piperine is obtained, and in latex of opium and morphine is obtained. Morphine is one of the potent analgesic that we use in surgical surgery, and in basically in used as an analgesic. So apart from this, alkaloids are also found in animals, like which animals and what are found. So we will study this. Like muscopyridine is obtained from musk of a deer, castoramine from North America rodent, Canadian beaver, Castor canadiensis, and from bacteria sources like phyocyanine from Pseudomonas aeruginosa. So in this, we basically study the distribution from which part the alkaloids are found, and from where, in which part, and in which specific plant or which whole plant, which active constituent found. We studied, and there are some active constituents which are found from in animals. So in this slide, we studied the parts where alkaloids are found. Now coming to the physical properties of alkaloid. Now how will identify it from physical properties? If something is alkaloid, or we will do the confirmatory test. So alkaloid are basically crystalline solid in nature. Few are amorphous solid, like example amethine, and few are liquid. So in liquid, there are non-volatile liquid which include iosine, pilocarpine, and volatile liquids include nicotine and conine. So alkaloids are solid in nature because presence of the oxygen atom in the ring structure. Oxygen is present in its ring, and which makes it solid. Most alkaloids are bitter in taste and mostly poisonous. So mostly alkaloids are bitter in taste and mostly poisonous, which also help in plant in their protection from insects and grazing animals. So they don't eat the plants because of its bitter taste, and they are poisonous also. Molecular weight of alkaloids is between 100 to 900 Dalton. They are generally colorless, but few are colored, like colchicine and berberine are yellow. Colchicine is one of the major chemical which we use in polyploidy. Like we convert like diploid plant to polyploid, like triploid or tetraploid, which increase the active constituent. Like in plant, when we use colchicine, it increases the tropane alkaloids. And salt of sanguine is copper red and bitterine is orange. Now, solubility and alkaloids are insoluble in water, but soluble in organic solvents. Exception include caffeine, cocaine, codeine, nicotine are soluble in water, and morphine, yohimbine are insoluble or sparingly soluble in water. So now coming to the isomerization, like we know that all the chemical compounds are like levorotatory or dextrorotatory. So alkaloids are optically active. Most of the alkaloids are levorotatory, but few are dextrorotatory. Example for dextrorotatory is conine, and few are optically inactive, like piperine. Optically active isomer may show different physiological activities, like L-ephedrine is more show more physiological effect than D-epinephrine. Now chemical properties. These are all about the physical properties, like what is the isomerization, solubility, and is weight, and is taste, solid nature. Now coming to the chemical properties. Alkaloids are natural organic compound. They contain basic in nature due to lone pair electron on nitrogen. So because of this lone pair, they are basic in nature. Exception include theobromine, theophylline, and amphoteric. They show both acid and basic nature. Alkaloids basically react with acid to form salt. These salts are usually freely soluble in water. For example, we gave quinine sulfate is water soluble, but quinine monosulfate is insoluble in water. And in alcohol, it is purely soluble and soluble and purely soluble in inorganic solvents. Exception including scopolamine hydrobromide is soluble in organic solvent. So in this we studied like the when alkaloid react with acid, they form salt, and salt is soluble in water or not. And it alcohol, how much is it soluble? It is purely soluble, sparingly soluble, or it is soluble in organic solvents. So this is studied up in chemical properties. So alkaloids may either way it exists as a free state as amine or as a salt with acid or alkaloid. So the main component of like or any alkaloid is the presence of nitrogen. So nitrogen exists as a primary amine like norepinephrine, secondary amine ephedrine, tertiary amine atropine, and quaternary ammonium tubocurarine. So basicity order of like we include like tertiary is most basic, then primary, then tertiary. Like weak base include caffeine. Strong base include atropine. Amphoteric nature shows by morphine, and natural alkaloid in colchicine. Colchicine, as we already told, like colchicine is used in the polyploidy. Like when we induce polyploidy, when we make plant from diploid to triploid to increase the tropane alkaloid content and other content also active constituent. So colchicine is used as a major chemical. Like uses, where is alkaloid used? Alkaloid is majorly used as analgesic, anti-malarial, anti-spasmodic, hypertension, mental disorder, and anti-cancerous. As we told, anti-cancerous, Vinca. Like what is the major function done by alkaloid in plants? So in plants, it protects against insect and herbivores. Like we told, like it is bitter and toxic, so plant doesn't. So animals doesn't eat it. It acts as a protective shield for plants. It is a source of nitrogen in case of deficiency. They are as an end product of metabolism or waste product or detoxification. And they are a storage reservoir of nitrogen for protein synthesis, which is needed in plants. They act as a plant stimulants and regulator in activities such as growth, metabolism, and reproduction. So in plants, they basically do the function like growth, metabolism, and reproduction. The major function also done by alkaloid is act as a detoxification agent. Basically, it removes the harmful substance. If it accumulates in the plant, it will cause damage. So it helps in the removal of harmful substance from the plant. Now coming to the classification, alkaloids are basically classified in four categories. First, in based on chemical structure, chemical nature and structure. Second, on pharmacological action. Third, in biosynthesis pathway, and fourth is taxonomical origin. So first, studying based on chemical nature and structure, it is further divided into three categories. Like first, on based on ring structure, monocyclic, bicyclic, polycyclic, heterocyclic, and non-heterocyclic. Second category is based on nature and basicity: primary amine, secondary amine, tertiary amine, and quaternary ammonium salt. This was studied in the previous slide. Based on Hagnot's classification is true alkaloid, proto-alkaloid, and pseudoalkaloid. Now coming studying the further this classification. Now first monocyclic. They basically contain a single and unfused ring like nicotine. As we can see the structure, one unfused ring is in the structure of nicotine. Bicyclic ring, they consist of molecules with 1,4 nitrogen rich cycloheptane structure. Example like atropine and cocaine. As we can see, the structure of 1,4 nitrogen bridge cycloheptane is seen in the structures. Polycyclic alkaloids, they have more than 2 rings. Examples: strychnine, cannabidiol, morphine, and codeine. As we can see from the structure, there are more than many rings present in the structure of morphine and codeine. Now, heterocyclic alkaloids. Like now we'll see the heterocyclic alkaloids. There are various type of alkaloids which contain these structures, like pyrrole structure in alkaloids which contain pyrrole structure. Example is like coca, from coca, and it is used as an analgesic. It is obtained from Arabidopsis coca family Aeroxylaceae. Second is pyrrolidine. Nicotine was obtained from tobacco from the family Solanaceae. It is used as a stimulant. Pyridine. Pyridine structure is present in the like lobila. Lobeline obtained from lobila family Campanillaceae, and lobila inflatae used in asthma. Piperidine. Piperidine obtained from black pepper, and Piperin nigrum is biological source and family is Piperaceae. It is used in anti-inflammatory activity. Imidazole. Imidazole is obtained from pilocarpine is active constituent, and it is obtained from pilocarpus, and family is Rutaceae. It is used in the treatment of glaucoma. Now coming to quinoline. Quinoline is the major constituent is quinine, treatment of malaria, and it belongs to family Rubiaceae. It is anti-malarial. Isoquinoline, like the other major active constituent is morphine, family is Peppervaceae, and biological source is Peppervaver somniferum. It is an potent analgesic used in surgery. Now indole, desirpine, active constituent from biological sources Rauwolfia serpentina, family Apocynaceae, and it's antihypertensive. Now purine, caffeine is obtained from tea, senescens, family Theaceae, and it's a CNS stimulant. Now from this classification, we studied like heterocyclic alkaloids and the structure present and what activity they show. Now non-heterocyclic alkaloids, like we can show like phenylethylamine. We obtained like ephedrine from ephedra. Biological sources Ephedra sinica, family Ephedriaceae, used in the asthma. Second is steroidal like conicine from curchi, family is Apocynaceae, and it is antidicentric. Third is tropolone, colchicine. As we studied in the previous slide, colchicine is used as a chemical for inducing polyploidy. So it is obtained from Colchicum autumnale, family is Colchicinaceae. It is used in the treatment of gout. Now the first classification is over. Now we will study the second classification. Second classification based on Hagar Test. They are classified basically on three categories. First is true alkaloid, proto-alkaloid, and pseudo-alkaloid. True alkaloid, they are alkaloid that directly derived from amino acid. They contain nitrogen in the heterocyclic ring. They are basic in nature due to presence of lone pair of electron or nitrogen atom. They give positive test for alkaloid. They occur either in free state as salt or nitrogen oxides. They are primary precursors of amino acid as L-arginine, L-lysine, phenylalanine, L-tryptophan, and L-histidine. Example includes guanine, morphine, and atropine. So this is the basic introduction which we studied in the previous slide, like why it is basic and it is give test positive test for alkaloids. Now coming to the proto-alkaloids, they do not have nitrogen in the heterocyclic link. As we studied, like true alkaloids have nitrogen, but they do not have. They are derived from L-tryptophan, phenylalanine, and tyrosine. They give positive test for common test for alkaloid. Although they didn't have nitrogen in their heterocyclic ring, but they give positive test. Examples include ephedrine, ordinine, and mescaline. Third is pseudoalkaloid. They do not derive from amino acid, but derive from the precursor of amino acid. They have nitrogen in their heterocyclic ring. Proto-alkaloid do not have nitrogen, but they have nitrogen in the heterocyclic ring. The nitrogen atom is inserted in the molecule at a late stage. They do not give positive test for common test for alkaloid. Like proto-alkaloids is giving the positive test for alkaloid, but they are not giving the positive test for alkaloid. Example include caffeine, conine, and capsaicin. Capsaicin is obtained from capsicum. Now, based on pharmacological action. Now, first two categories are done. Now, the pharmacological action. So, narcotic analgesic. Narcotic analgesic include like opium and tea from Papaver somniferum and tea sinuses. They belong to a family like Papaver and Theaceae. Example, active constituents are morphine, codeine, and caffeine. They are narcotic analgesic. CNS stimulant include nux vomica, logenaceae, strychnine. Anti-cancer include Taxol and Vinca. Vinca we studied in the first slide, like Vinca, it is anti-cancer. Two active constituents are obtained from Vinca, vincristine, vinblastine. Taxol is belong to family Taxaceae and Vinca from Apocynaceae. And now coming to antihypertensive. Antihypertensive, Rauwolfia from biological source, Rauwolfia serpentina from Apocynaceae and Reserpine. Bronchodilator includes ephedra and Vasaka, ephedraceae and acanthaceae, ephedrine and vesiconone. Now, smooth muscle relaxant. It includes two drugs basically: belladonna, atropa belladonna is the biological source, opium, and the Papaver somniferum is the biological source. The atropa belladonna belong to the family Solanaceae, and opium belongs to the family Papaver. And from atropa belladonna, the active constituent is atropine, and opium is piperine. Antitussive. It is also obtained from opium, Papaver, but the active constituent is codeine. Atropa belladonna is atropine. It is acting both as a smooth muscle relaxant and medreactive. Myotics, pilocarpus, belong to family Rutaceae and active constituent pilocarpine. Antiparasitics, Shincona, quinine, treatment of malaria, IPAceae, Rubiaceae, and the active constituent is amytine. Local anesthetic, coca, Erythroxylum coca, family Erythrolaceae, and the active constituent is cocaine. Anti-arrhythmic, Shincona, and the family is Rubiaceae, and the active constituent is quinidine. Now, based on synthetic pathway, as we know, whether every drug, every chemical compound follow a certain pathway. So, on the basis of pathway, these drugs are classified. These compounds are classified. So, the first is ornithine derived. First is pyrrolidine, example nicotine, and tropane, example atropine and cocaine. Lysine derived include piperidine and pyridine, and quinazolotine. Example is conine, lobeline, and lupinine. Tyrosine derived include isoquinone and amino. It include morphine, codeine, berberine, colchicine for the inducing polyploidy. Tryptophan derived include indole and quinoline. Indole include ergot, vincristine obtained from vinca, vinblastine, reserpine, strychnine. And quinoline include Shincona, quinine, and quinidine. Histidine derived include imidazole, and the example is pilocarpine. Piline derived include amino alkaloid, and example is ephedrine. Now, the last category and is include like based on taxonomical origin, where it is found. Like alkaloids are classified on the basis of biological source. Like we gave the example like quinine from bark of Shincona calicea, Rauwolfia from roots of Rauwolfia serpentina, and morphine from dried latex of Papaver somniferum. So now include the general chemical tests. Like there are various chemical tests which we identify, like it is a like alkaloid or not, or if any in any substance or any compound, any liquid, any solution, if alkaloid is present or not. So there are major famous tests for alkaloids like include drag and drop test. In this, potassium bismuth iodide solution, also known as drag and drop reagent, plus alkaloid sample is mixed and orange PPT is observed. If orange PPT is observed, that test is positive, alkaloid is present, otherwise not. Second is Mayer's test. Potassium mercury iodide solution, also known as Mayer's reagent, plus our alkaloid sample, then cream PPT is obtained. If not, then alkaloid is not present. Wegener's test, iodine potassium iodide solution, also known as Wegener's reagent, plus alkaloid sample, and reddish brown PPT is obtained. And if not, the alkaloid is not present. And last is Hagar test, saturated solution of picric acid, also known as Hagar's reagent, plus our sample, yellow PPT if observed, alkaloid is present, otherwise not. Now these are the specific reagent tests. These are done for the specific alkaloids like Vitali Morine test. Alkaloid, for example, propane plus Vitali Morine agent, which is fuming HNO3 plus alkaloid KOH solution are mixed. If violet color is open, this test is positive. And the second is Van Urk's test. Alkaloid sample, ergot alkaloid, that is indole, plus para-dimethylamino-benzalide is mixed in dilute solution of H2SO4 and some traces of FeCl3. Blue color is observed. In thalloquine test, Shinkona powder, quinine alkaloid, plus bromine water, plus dilute ammonia solution is mixed. Emerald green color is observed. In muroxide test, alkaloid sample, purine derivative, that is caffeine, plus KClO3, plus HCl drop is evaporate to dryness. And then a dried residue is mixed with ammonia vapor. If purple color is observed, then it shows the presence of alkaloid in our sample. So concluding all the things in our lecture, so we studied about what is alkaloid, where it is found, its categories, its physical and chemical properties, its uses, and finally its chemical test. So I hope you get all the idea about alkaloids and its all the basic idea. So ending now, like this is all from my side. So thank you.